This is what's known as a Punnett square. Punnett''S. A Punnett square is used to show the possible offspring for two parents. And in this case, it's the simplest form of a Punnett square because it only shows one trait and the two potential alleles for each trait. Now, Punnett squares can be quite large. In a situation where there's two traits being investigated, the Punnett square has 16 boxes, so that each time you add a trait to look at, you are increasing the size of the square exponentially. Anyway, in a Punnett square like this, you generally have one side represents the mother and one side represents the father. And this is the genotype of the father divided into its two alleles or two gametes. One of his gametes will have a big B and one can have a little b. So he's heterozygous for that particular trait. And then the mother has two small b's. So she has two gametes. Each gamete is the same in the fact that it has a little b for this trait. Now when you select the particular letter, you can use a letter that reminds you of the trait that it's investigating. However, you use the same letter whether it's a capital letter or a small letter, regardless of the phenotype. For example, if this represents hair color and you want big B to represent brown, then little b can be red, but you do not change the letter to something else like R. In other words, it has to be the same letter, and that reminds you or keeps straight in your mind the fact that the two gametes are in the same trait. The second point is that when you select a letter, the capital letter is the dominant and the little letter is the recessive. And for each situation, you combine the big B, you combine one allele from the father with one allele from the mother. And whatever's in this box represents this father and this mother allele, or this father and this mother's gamete. And so this is going to be the genotype for one offspring. Then here you have this one with this one, so this is going to be the genotype for that offspring. You have this one with this one, so this is the genotype for that offspring, and then this is the genotype for that offspring. And so the purpose of the square is to keep all this straight, so you can see that two offspring would have the same homozygous recessive genotype, and two offspring would have heterozygous dominant offspring or genotype. These two would be brown-haired, these two would be red-haired, and it would be 50%, if asked for a percentage. 2 out of 4 is 50% homozygous recessive and 50% heterozygous. 50% brown-haired, 50% red-haired. So with that, let's take a look at some problems. And I will use a notepad here to show you how to work these problems. Let's read the first problem. Let's say that in seals, the length of whiskers is controlled by a single gene with two possible alleles. Now I've gone ahead and decided to label these for you. Capital W represents long and little w represents short. And it's actually conventional a lot of times to pick a letter that doesn't rhyme with either possibility so that you're not tempted to try to change the letters. What are the 4 possible genotypes of a cross between a homozygous long and a heterozygous long? And then what are the phenotypes? Okay, so I've already done this over here. Here's the homozygous long individual, mother or father, and they have 2 capital W's. Here's the heterozygous long, a capital W and a little w. So when this combines with this, you have 2 capital W's when this combines with this, you have two capital W's. When this little w combines with this big W, you have a big W and a little w. And similarly, when this big W combines with this little w, you end up with a big W and a little w. So half of the offspring are big W, or homozygous long, and half of the offspring are big W, little w, or heterozygous. It's not wrong, by the way, to say heterozygous long. All of the offspring, because big W is dominant, all of the offspring will have long whiskers. So 100%, all four have long whiskers. 2 out of 4 have heterozygous genotype. So 50% are heterozygous genotype and 50% are homozygous long genotype. The second problem is over here. In purple people eaters, the presence of a horn is controlled by a single gene with 2 alleles. And this time I did pick a letter that was consistent with at least horn. So if capital H, the dominant, is a presence of 1 horn, and little h, the recessive, is no horns. So what are the genotypes of the 4 offspring if 2 hybrid, and the term hybrid means heterozygous, horned parents? What are the genotypes of 2 heterozygous horned parents, and what are the phenotypes? And I've done this right here. Here's the genotype of each parent. They're hybrids. In this case, they're hybrid. And so you have a big H and a little h for the mom, and a big H and a little h for the dad. And each contributes one of those 2 along the side here. This is dad over here, and this is mom down here. So you have big H plus big H is big H. Big H plus little h is big H, little h. Little h and big H here is big H, little h. And then little h and little h leads to 2 little h's. And the genotypes, you have 1 genotype here, 2 genotypes that are big H, little h, and 1 genotype that's big H, little h, little h. So you have 1 of these, 2 of these, and 1 of these. So the percentage is 25% homozygous long, or horn, 50% heterozygous, and 25% no horn. In terms of phenotype, because the presence of a horn is dominant, you have 3 out of 4 that'll have a horn, and 1 out of 4 that has no horn. And look over this fairly carefully, because we will have something like this on a quiz or a test. It's important that you know this. Now moving on, this is what's known as a pedigree. A pedigree is a diagram that follows a particular phenotype through several generations. Now let's go over some conventions for phenotype, or for pedigrees. First of all, the squares are males, the circles are generally females, and when they're joined by a line, it means that they're married, they're having children, they're mates. And these are their children. Anything connected to this line is their children. So these represent their children. And open, or white in this case, is an unaffected, for whatever the trait is, it's unaffected male, unaffected female. It can also say represent a brown-haired male and female, and then blue would be blonde hair. It's usually the less common phenotype is a different color. And here's 2, say, blonde-headed individuals. So these 5 children, 3 are unmarried, 2 are married. Here's a female that's married and a male that's married. This male has 1 child that's a male who's also married, and they have 5 children, 3 boys and 2 girls, 2 of which are blonde. And these 2 are married. They have 3 children, none of which are affected, 1 male and 2 females, and this one is married. So you have 4 generations. Now let's take a look at a problem and we'll go through it the same way we went through the last one. We'll start down here. The diagram below shows a pedigree for a genetic characteristic, color blindness. What is the mode of inheritance? And what is the genotype of each individual? Okay, the mode of inheritance means, is it autosomal dominant? That means, is the color blindness dominant? Is color blindness recessive? And when we get to sex-linked, you can also have sex-linked dominant and sex-linked recessive, but we're not talking about sex-linked yet. And then the second question is, what is the genotype of each individual? Okay, so you can probably figure out each individual. Now, the way to tell in regards to the mode of inheritance is to look at who is affected. So we have a female, a male, and let's just assume that they colored white is unaffected males and females. So these are unaffected or color normal vision, and the colored squares are affected individuals. So we have an affected female marrying an unaffected male. They have 1, 2, 3 children. This child is affected. These two are unaffected. Now here's an interesting clue. You have 2 unaffected individuals, but they have an affected individual in their family. And here's an affected person married to an unaffected person, and all 3 of these are unaffected. Okay? So if you have 3 individuals, you know that the 2 possibilities are autosomal or non-sex linked dominant. And what that means is that if their genotype is, let's just use C's for color. So capital C means normal and little c means not normal. If they're autosomal dominant, all the affected individuals have to be either 2 capital C's or 2 little c's. And the normals are only 2 little c's. You also know that if it's recessive, then if you use big C here as a dominant, now remember we have to use the little c as recessive now. So that means these are colorblind individuals, and this is the non-colorblind individual. It reverses if you think it's recessive, so that big C's and big C little c are normal, and 2 little c's are not normal. Now, if this is the situation, that means that either of these, the affected individual is going, in this case, the affected individual is going to affect all of the offspring. In this case, the affected individual is going to affect 2 of the 4 offspring. And here you have a situation, so there's no situation where an affected individual doesn't impact anybody. And so you have right here a generation where nobody is affected. So that it's very unlikely that this is a dominant situation. It's much more likely that it's recessive, which means that in the first generation, this first person can be either cap C, or cap C little c, and the affected female has to be 2 little c's. But now if this guy is cap C, he has to pass that to all the children. He has no choice, so he can't have an affected child. So his only option is this. So in the first generation, the male is cap C little c, and the female is little c little c. The second generation, this person again can be either cap C little c or cap C, but can't possibly be cap C because mom is affected, so she had to pass a little c to this girl. So the fact that she's not affected tells you that she is cap C little c. This guy, similarly, again, has to receive a little c from mom and a big C from dad. So this male child has to also be cap C little c. This guy right here has to have two little c's. So in that second generation, the male is cap C or little c. This one is cap C little c, and this is cap C little c. And these are all have to be this way. To go to the next generation, now the fact that you have two unaffected parents, and you know that the female here is cap C little c. If you have an affected daughter here, that means this guy had to pass a little c to him as well. So she married a, so that male also has to be cap C little c in order to have a child that's affected. And this child is little c. This child, on the other hand, could be either cap C, getting a cap C from each of these parents, or a cap C little c, getting a cap C from one parent and a little c from the other parent. You can't be absolutely certain about that. These two children, this one we know is cap C little c in the second generation. So what about this one? Well, the fact is that they have two normal. They're normal, and these two are normal. So she could be two cap C's, and there's no subsequent to tell you anything. So you can't know whether she's also cap C little c or cap C. They've only had two children, so there's not enough children to get the percentages involved, and so forth. I'm going to stop at this point for this example, and when we come back, we're going to talk about looking at two traits instead of just one. So something a little bit more complicated. And we can go over a problem like this in class as well if this has been in any way confusing to you.